Hello everyone, my name is Jiajia Yang. My presentation title is Layer-Specific Finger Representations in Human Iris 3B. This is a collaboration between Ohio University, National Institutes of Health in US, and the National Institutes for Physiological Sciences in Japan. I have nothing to disclose for this presentation. First of all, I will show you some basic information of the somatosensory system. So there are four types of mechanoreceptors in the skin, and they can be categorized into two groups. The first one is named as slowly adapting receptors, SA, and which are sensitive to the special features of the object. The second one is named as rapidly adapting receptors, RA, which are sensitive to the temporal features. So both SA and RA receptors will be active at the same time to encode the information which contribute to our tactile perception. So when we touch an object, and the information encoded by these receptors will be projected to the primary somatosensory cortex S1, which is located on the postcentral gyrus. Typically, the area of the body corresponds to a point on S1, which is called the somatotopic organizations. S1 can be divided into four sub-areas, 3A, 3B, 1, and 2. And the somatotopic maps in each sub-areas are different, since the receptive field of neurons in each area are different. The previous animal study showed that the receptive field of area 3B are small, so we expect to find a clear finger representation within area 3B. The receptive field of area 1 and 2 are becoming larger, which reflect a convergent input from area 3B. So in other words, the finger representation in area 3B are clear, and area 1 and 2 may be not so clear as in area 3B. Using fMRI, we would expect to find a clear somatotopic map in area 3B, and the map will become blurred in area 1 and area 2. So in our previous study, we tried to map the 4 fingers area in human S1 at 3 Tesla. So we poke in subject the fingertip using a stick and scanner. And here's the map of one subject. We can see that even in area 3B, finger activation area are overlapped, and it is difficult to isolate one from other fingers. So this result did not match the receptive field in S1 as we have mentioned in the first slide. And one possible reason for this result is the low spatial resolution at 3 Tesla. However, the overlap is still there when we look at the result at 7 Tesla with 1.5 millimeter resolution. For example, in this study, in area 3B, the borderline between fingers are not clear, and some of the areas are active by all fingers. So beyond the resolution issue, some sort of cross-finger interactions or feedback modulation across these areas may be the factor for the overlap. So the finding from one recent study at 7 Tesla support this view by found that attention modulated somatotopic maps, but they did not discuss too much about the mechanism of this attention modulation. And layer functional have the advantage to test cross-finger interaction as well as feedback modulation across these areas in S1. For example, in our previous study, we found that finger input activated the middle layers of the area 3B, where the upper and deep layers are more likely to active by the predictive feedback. So we also found that prediction error feedback signal may modulate the activity in deep layers of area 3B. So in this study, we like to look at the layer-specific activity of each finger in the sub-area of S1, and we hope to provide some insight into understanding the tactile hierarchical processing pathway in human somatosensory system. So we designed a metal-free device, and you can see from this video, when the operator presses one side down, and the other side will rise up to stroke the subject's fingers. So we use the multi-contrast Vaso-Bolt sequence, which is a similar one that has been published in Renzo's paper. And the spatial resolution in depth is 0.71 millimeter, and the slice thickness is 1.8 millimeter. The slice is perpendicular to the front bank of postcentral gyrus. A total of 12 slices were recorded, and the effective TR is 3.49 seconds. Basically, this is an interleaf acquisition mechanism, and each single visual image is interleaved with the body image. We separate them, and then we did pre-processing using fMRI, SPM, and FSL was used for statics analysis. And here's the body map of 4 fingers in S1 for one subject. You can see that the 4 finger activations were organized in a somatotopic manner in area 3B, but not show clear patterns in area 2. And here are the body color maps of index and middle fingers from 10 subjects. And you can see some individual difference, but all subjects show the same finger arrangement along the area 3B. But to see the finger distribution from both columnar and layer dimensions, we have to define finger mask for each area based on these results. And here is an illustration of S1, and the blue box indicates or scan volume. So after drawing the borderline of green and white matters in area 3B, and we calculated the layers using linear package. So in each finger region, the peak response occurs in the middle layers. And we also found a clear finger representation in middle layers of the 3B, and rather than that in the upper and deep layers from the visual profile. In contrast, the both activities showed a mostly similar pattern across all layers. To see detailed data in area 3B and area 2 from layers and common direction, we then generate a new mask set that look like this. To give you a quick image, here I plot the data from one subject. So after repeat this process to all subjects, we normalize the data point in column direction. And here are the average body profile, and I removed the error bar from the figure for visualization. And each small graph in this figure represents one depth data point covering all columns. This figure confirms the clear activation peaks for each finger in area 3B and a double peak pattern in area 2. But the activation pattern seems to have no difference across layers. And here are the visual profiles. I found that 4 finger peaks in area 3B and the double peaks in area 2, and are more like in the middle layer rather than in all layers. And we are trying to explain our results using the previous animal finding from the basic science side. As shown in this model, we know from animal studies that the first order sensory information of each finger will be projected to the finger area in 3B. Then these signals will be projected to area 1, 2 for further processing. In area 3B, we also know that the sensory signal from cell mass will be projected on the middle layer of each finger areas, and then spread it to both upper and deep layers. So this is the reason why we found the clear finger maps in the middle layers. To date, however, no direct evidence allowed us to model the layer-specific information flow across fingers, and it is more challenging to model the information pathway from 3B to area 2. And now we are trying to discuss the underlying mechanism of these layer-specific finger maps. And for more detail about our study, please stop my poster, and I would be very happy to talk with you. And finally, I would like to thank to all my wonderful collaborators and thank you very much for your listening.